Antibodies are produced by particular white blood cells in response to an invading pathogen. When your body comes into contact with a pathogen it doesn't recognise, it triggers particular white blood cells to manufacture antibodies. Scientists have been able to extract the particular white blood cells involved after a specific immune response has occurred and use them to produce the single antibody that can destroy the pathogen. This single cultured antibody is called a monoclonal antibody. So how do we get large amounts of different monoclonal antibodies for different purposes? The white blood cells that can make specific antibodies for specific diseases are all a type of white blood cell called B lymphocytes. In the laboratory, mice are used to provide the B lymphocytes. The mouse is exposed to materials that will cause its white blood cells to produce antibodies. The white blood cells are then extracted from the spleen of the mouse. But there's a problem. These extracted B lymphocytes won't survive outside the body for long and won't divide. However, we know that cancer cells have the ability to continue to divide and divide without stopping, and this is why they form tumours. So scientists had the clever idea of mixing the extracted B lymphocytes with some cancer tumour cells. The mixing process means that some of the cells fuse or join together to produce a new hybrid cell called a hybridoma. These hybrid cells will produce antibodies and will keep dividing. This means that they'll multiply to produce copies or clones of themselves indefinitely.
So now we have lots of hybrid cells all capable of producing antibodies and of multiplying indefinitely. But what we want are separate groups of cells with each group producing a different antibody. To get this, the hybridoma cells are separated into single cells and are left to form identical copies of themselves. These copy cells are called clones. Each group of clones is then tested to see which antibody they're producing. The antibodies build up on the gel that the hybridoma clones are growing on. The antibodies are easily separated from the cells and gel and then purified. This gives us a single pure antibody produced from clones of one cell. This is how monoclonal antibodies get their name. Mono means one and clonal means from identical cells. The production of these specific antibodies is incredibly useful and has a huge potential for medical research. Imagine these antibodies as arrows, but instead of having a pointed arrowhead at the end, each type of arrow has a different shape of socket. So if you want to find something that's square, you fire off arrows with square sockets. Once the arrows have found the square thing you're looking for, they fit onto it and stay there, pointing out where it is. This is how the antibodies work. They're very specific and search out in the body the specific chemical they're matched to.
A pregnancy test is a simple way of using a monoclonal antibody to point something out. When a woman is pregnant, she produces some extra chemicals that non-pregnant women don't. A pregnancy test kit contains a monoclonal antibody that fits the chemicals produced in pregnancy, so you can perform a simple test. If the woman is pregnant, the antibodies fit around the pregnancy chemical and point it out. If she isn't pregnant, there's no chemical for the antibodies to bind to or point out, so you get a negative result. Monoclonal antibodies could be used to target and identify, even neutralise unwanted substances in the body. For example, to prevent the consequences of a drug overdose. If you could inject specific antibodies that would quickly identify, bind to and neutralise the drug, you could stop the potentially fatal effects and improve the chance of the patient's recovery to full health.
Important research is going on at the moment in the use of monoclonal antibodies to treat cancer. Scientists are trying to attach poisons to antibodies which match antigens on specific cancer cells. The hope is that the antibodies will attach only to these specific cancer cells and so deliver the poison directly into them. This would mean that specific monoclonal antibodies would target specific cells to label or destroy them and not harm non-cancer cells. This would result in far fewer unpleasant side effects than the current use of drugs and radiotherapy in cancer treatment. Monoclonal antibodies are already very important in science and medicine. New ways of using them is an exciting and rapidly expanding field of scientific research.